So the topic is biotech application. So what is biotech application? So first thing is what is biotech or biotechnology? In biotechnology, through different technology, bio means living. Technology means through different techniques we produce new products, new methods, or modified organism. So what is new products? New products like one of the example coronavirus vaccine, insulin, these are the new products. And the different methods like DNA isolation, microscopy, and difference process so that we can work through biotechnology. Next one is new modified organism. So what is new modified organism? That is, we change in gene sequence of organism. That means if this is an organism and this contain a DNA, change in sequence of gene or we adding a foreign gene. Here this is a foreign gene added to a particular specific organism is known as new modified organism. Through this new modified organism, we producing different enzymes or different microbes, modified microbes that helps in different biotechnological process. So what exactly I will discuss all the things. Three critical research areas of biotechnology. These are first one is providing best catalyst. One is providing best catalyst. That means we producing proper pure enzymes. We know the function of enzyme that helps in activating or enhance the activity of any chemical reaction or any reaction. So these enzymes helps in different reaction. Next, genetic modified microbes. These microbes help in different function. How these microbes help in different function, I will discuss later through this application of biotechnology. First point, extraction or producing best catalyst of enzyme. Second one, creating optimal condition. The meaning of optimal condition, optimal indicates suitable environment. Suitable environment. So what is suitable environment? Let's think, when you're producing different types of enzyme or proteins, to producing different types of enzymes or proteins, it need to maintain a proper pH, proper temperature, proper pressure, proper chemical component, not more than or not less than H required, as we need or providing suitable conditions. And the third one is purification of proteins. So how did the purification of proteins? So first, one of the example, if I'm discussing here, this is a test tube and this test tube contain blood. We know blood contain two types of part. One is called plasma and another is known as corpuscles. So what is plasma? Plasma contain different proteins. One of the example of protein is known as albumin. These albumin are extracted and then purified. This purified protein helps in different function. So this is known as purification of proteins. So first one is providing enzyme, a best catalyst. To maintain this catalyst work on a proper condition, we're creating a optimal condition or suitable environment. And the third one is purification of proteins so that we can do the things we need. Then the application of biotechnology. This is shortly I am discussing this thing, and according to our NCERT, I am discussing all the things that is properly mentioned. So first one is a diagnostic. So what is diagnostic? Diagnostic means to detect a disease. Through biotechnology, we detect different types of disease, and this disease mostly recognized by proper doctors using different biotech tools. One of the example, a blood test developed, a blood test developed through biotechnology that measure low density lipoprotein or bad cholesterol. And second one, in case of some cancers, that one of the example, prostate and ovarian cancer, we through blood sample we detect these cancers. So these are the application of biotechnology in diagnostic, and this detect and diagnose with a faster rate than normal tools and techniques, because we are using different biotechnological tools. So next one is biotech in therapeutics. So what is the meaning of therapeutic? Therapeutic means it is a branch of medicine and that helps in treating diseases. So one of the example of this therapeutics, or in case of biotech application in therapeutics, we producing monoclonal antibodies, different type of antibodies, recombinant vaccine. So what is antibodies? Antibodies provide immunity in our body. Recombinant vaccines means through different types of genes to combining and we producing, let here is one gene, this is one gene, and this is one gene, combining these two genes producing a new protein or new antibodies. So as like recombinant protein, stem cell and genetic therapy, we using biotech in therapeutics. Next one, genetically modified crops for agriculture. This genetically modified crops means that this is a organism and this organism contain DNA. This DNA maybe changes the sequence of, let's consider this sequence contain A, T, C, G or something. We change the sequence of DNA and modifying this gene to producing new protein, or in second condition, this is a gene after we adding a new foreign DNA. We adding a new foreign DNA, and then after we producing a new or introducing a new gene to producing new variety of protein. So this is known as genetically modified crops or genetically modified organism, that is application in crops. Second is after that processed food. So biotechnology in bioremediation. So what is bioremediation? Using different microbes and bacteria to decontaminate affected areas. So mostly during different companies production of different contaminated like junkyards, industrial spills, land development and different fertilizer. These are contaminated. This contaminated are digested, or this contaminated are removed through different types of microbes and bacteria. This is known as bioremediation. Next one is the application of biotech in processed food. So what is processed food? This biotech helps in it increase the edibility of food, that is like vegetable, fruits. So it enhances the edibility and it increase the consistency. And third one is it enhances the shelf life of food. That means it helps in storing for long term and preventing growth of unwanted toxin so that so that it shows a edibility, consistency, and shelf life of food. That means it is so for a or store for a long term, and it helps in production of antimicrobial agent that kill different other undesirable microorganism. Next application is biotech in waste treatment. So what is waste treatment? So using different methods like physical, chemical and biological. Mostly I am discussing biotechnology, that is biological method to treat different types of pollutants using or introducing different types of microorganisms, enzymes or plants to remove or reducing different solids, organic matter, nutrients, pathogen, toxic substances using these microorganism, enzymes and plants. So this is the application of biotech in waste treatment. Now, application in energy production. So mostly production of biofuels. One is example biomethane, biodiesel. These are the example of biofuels known as biofuels, and this is produced through the technology of known as biotechnology. These are the major application of biotechnology in different field. Now I am discussing according to our NCERT. The first one is known as that is the biotechnological application in agriculture. So first I am discussing why we using biotechnology in agriculture. The main reason behind to using the biotechnology, that is increasing the food production through using or during using we producing quality plant, quality food, nutritive food. So this is the application of biotechnology in agriculture. So it contains different parts like agrochemical based agriculture. It contain, first I am discussing agrochemical based agriculture, secondly what is organic agriculture, and thirdly genetically engineered crop based agriculture. So first one is agrochemical based culture, secondly organic farming, and thirdly genetical modified organism. These are first agrochemical based culture. So agrochemical based culture, it is through by chemical fertilizer. You using we using chemical fertilizer to develop this agromens crops. We using chemical fertilizer helps in increasing the yield of crops, and secondly using chemical pesticides, so that it has after some year it shows different disadvantages like decreasing the fertility of soil, harmful for living organism, for their cell and body living tissue. So we want to develop, we want to further process on organic farming. So this organic farming to production different organic vegetables, organic fruits, we using organic biofertilizer like different types of neem coated, different types of plant based to produce fruits, vegetable and these things, using biopesticides, using bioinsecticide. The main disadvantages of this, more expensive. These fruits and vegetables are more expensive than chemical based agriculture. And secondly, production costs are very high. Why? Because we cannot produce organic fruits or vegetable in a normal condition. So it need to a proper condition. One of the example polybag, like polybag and another things. So another is smaller amount of production. The production amount is smaller amount. In case of agrochemical, this is produce money much more amount or production level very high. But here is the production level lower than agrochemical. And susceptibility to illness, easily ill due to different microbes, but here we're using different chemicals so that it is not easily affected by microbes. So further we goes into genetically modified organism because we want to overcome this disadvantages that found in agrochemical and organic farming. And this genetically modified organism are mostly producing through transferring different genes from one plant to plant or plant to animal or plant to microbes, is like we transfer of different gene or introducing different foreign genes in a particular plant, animal or microbes to produce genetically modified organism, and this is genetically modified crops. Now we're producing through different introduction of genes. The first one is that is enhanced nutritional value of food, that is known as golden rice or vitamin A. So what is golden rice and vitamin A? One of the example, the application of biotech in crops, that is producing golden rice. This is a normal rice and this is a golden rice. The golden rice that produce vitamin A more than normal rice, and the application of vitamin A that helps in maintenance of body. One of the example, that is protect from night blindness. So this is the application of vitamin A. Now, through biotech, how we produce this golden rice. One of the plant known as daffodil. This daffodil contain beta carotene gene and produce beta carotene. This beta carotene is the derivative of, or vitamin A is the derivative of this beta carotene. So we need vitamin A in golden rice or in rice, that is for different function. So firstly, this gene are isolated, then introduced into one bacteria. This bacteria named Agarobacterium or any bacteria. Thus after insertion into bacterium, this bacterium contain now this gene. This the red color is the gene, introduced gene of daffodil in the bacteria. This bacteria then transfer into a rice plant. This is a rice plant. This is a rice plant. After transfer into rice plant, it produce, or in after transfer it integrated with plant and produce beta carotene. So this beta carotene produce vitamin A. So now we call it golden rice. So this is the procedure of production of golden rice, and this is the application of biotechnology. Next, to reduce post-harvest losses, flavor saver tomato. So in flavor saver tomato, we reduce post-harvest losses. So what is post-harvest losses? Means that normally after some time the tomatoes are degrade due to presence of ripening genes. But when we through biotechnology we inactivate different ripening genes, so this tomato not easily ripened and stored for a long term. And this is a great application of biotechnology. The third one is to increase tolerance against abiotic stresses. So what is the meaning of abiotic stresses? These are cold, drought, heat and salt. So we using genetically modified or gene to introducing different genes to tolerate these plants from cold, drought, heat and salt. Now, another is to produce biopharmaceutical products like hirudin in brassica naples. We extracting or producing hirudin in brassica naples. This hirudin is known as anticoagulant factor. This hirudin is found in brassica naples. Now, to produce weedicides. Weedicides means when we using, let's consider this is a plant, this is a productive plant, and this is a weedicide, weeds. When you're using some chemicals for weeds, this plant are normally, these plants are normally degrade or destroyed. But when you using weedicide, weedicide through genetic modified plant, only these weedicides are destroyed, not the productive plant are destroyed. Another is increased efficiency of mineral use of plants. This reverts early exhaustion of fertility of soil. Means the mineral use that helps in this revert early exhaustion of fertility of soil. Another is to reduce reliance dependence on chemical pesticides, products of pest resistance plant, insect resistance plant. Application of biotechnology in agriculture. So insect resistance plant. So what is insect resistance plant? So through producing insect resistance plant, we using an bacteria. This is known as Bacillus thuringiensis. This Bacillus thuringiensis introduced in plant and crops are the best example of insect resistance plant. This BT plants or Bacillus thuringiensis contain a specific gene. This gene known as cryogens. This cryogen is present inside the bacteria known as Bacillus thuringiensis. This cryogen then inserted, let's consider this is a cryogen. This cryogen then inserted into the plant cell, then it produce cryoprotein, produce cryoproteins. These cryoproteins act as insecticide, pesticides, and how these are act and how will inject these genes I'll discuss. So first, Bt toxin gene. This is the Bt toxin gene or cryogen plus in plant cell that is produce plant contain also gene. This gene integrated with this toxin gene. This is the plant gene and this is Bt gene or cryogen. This cryogen produce cryoprotein and the combination of this one gene and this one gene is known as recombinant gene. So through this cloned from bacteria and expressed in plant to provide resistance to insecticide. Through we protect from insecticide and biopesticides. It is called biopesticides because through biotechnology, not using any chemical, we protect from insecticides. So it is called biopesticides. In application, we producing Bt cotton, Bt corn, Bt rice, Bt tomato, Bt potato, Bt soybean. So in our NCERT, it is given only about Bt cotton. So before I am discussing what is Bt cotton, first I am discussing Bt toxin. Bt toxin gene is Bt toxin gene is coded by cryogene. And this cry IAC, cry IAAB that control cotton ball ohms, that control cotton ball ohms, and this cry IA control this control kern borer. So this is the application of Bt toxin gene in different organism, different plant, and different against different insects. So next, Bt cotton. This is mentioned clearly in our NCERT, the example of Bt cotton. So Bt cotton is some strain of Bacillus thuringiensis produce protein. This protein kills certain insect. So for protein like Bt cotton gene protect from lepidopterans. These are tobacco boredoms and armyworms. And the coleopterans, these are beetles, and these are protected by Bt genes. Diapterans, these are flies and mosquitoes. The one important point here written, the choice of gene depends upon the crop and the targeted pest. Depend upon the crop. It may be which type of crop it depend. And second is which type of targeted pest like lepidopterans, maybe coleopterans, maybe diapterans. According to gene and according to plant, as most Bt toxin are insect group specific. So it is insect group specific. So this is the mechanism of Bt protein or Bt toxin against 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 insect. So first one is Bt, Bacillus thuringiensis. This Bacillus thuringiensis producing a crystal protein. This crystal protein is a toxin and that is act against insect. Now this toxin in the form of prototoxin that is inactive, but this gene I'm discussing little bit here. This gene, this is cryogene or Bt gene. This gene now transfer to the plant. Now transfer to the plant, and now this plant produce Bt protein. Let consider this is leaf and this leaf surface producing Bt protein. So when this Bt protein producing plant due to insertion of this cryogene or Bt gene or Bt toxin gene. After insertion into plant producing Bt protein, when any insect come to eat this leaf, then it enter the toxin inside the insect, so that inside the insect it contain alkaline pH. Due to the alkaline pH of gut, this protein activate, and this activated protein bind to surface of mid epithelial cell. Let's consider this is stomach, this is the gut lining, this is gut lining. This gut lining now activated protein enter here and pore, that create pore in the cells, so that if create pores, cell lysis happen. Due to cell lysis, cell death happen. So the insect dies. This is the mechanism of action of a protein against this insects. So this is the same discussion. This is cryogene. This cryogene produce cry-mRNA, cry-mRNA. This from mRNA producing protein. This is called Bt protein. This Bt protein is inactive in the inside Bacillus thuringiensis, and this bacteria transfer to the plant when any insect or something that ingest ingest this protein shows death. This is the application of Bt protein against this insect. Next one is Bt nematode resistance plant. Next one is Bt nematode resistance plant. So what is Bt nematode resistance plant? One of the example nematode Meloidozin incognita that infect the roots of the tobacco and cause a great reduction in yield. So one of the example of nematode resistance plant is Meloidozin incognita that infects the root of tobacco and cause a great reduction in yield. To prevent this nematode strategy adapted, this is known as RNA interference technology. This RNA interference technology helps in gene silencing. So what is gene silencing? Means genes are not expressed. If no expression, no proteins. No proteins. So RNAi takes place in all eukaryotic organisms as method of cellular defense. The main function of this RNAi, that is cellular defense. Once again I am discussing. So the nematode name Meloidozin incognita, and that infect root of tobacco plant and cause a great reduction in yield. To prevent this nematode using RNAi, that is RNA interference technology. RNAi is a process of gene silencing. It is a process of gene silencing, and this RNAi takes place in all eukaryotic organisms as method of cellular defense. Remember this, RNAi act as cellular defense. So mechanism of RNAi, how this RNAi work, I'll discuss. So this is the artificial target gene of nematode. Gene of nematode. So this nematode are targeted by RNAi technology. Through RNA, we silencing the genes of nematode. After silencing this gene, it not produce toxin automatically. Due to no produce in protein, it will destroyed or degrade or eliminated or end of this life of this nematode. So how this nematode are targeted by RNAi. The RNAi bind with gene of nematode and silencing this gene. So no expression. No expression means no protein. No protein means organism dies. So here how we producing this or how we silencing this gene of nematode. The first we producing two genes artificially. This is one, this is two. This artificial gene one contain during transcription. We know genes contain promoter, transcription and structural gene and also structural. But here is antisense, here is sense. In antisense, promoter first, in sense termination fast. In antisense, termination last. In sense, promoter last. So this produce a mRNA. Antisense produce a mRNA and sense also produce a mRNA. But the difference is, let's consider this mRNA contain AUG. Only imagine this contain AUG. Then this contain, in case of A, this is U, in case of U, this is A, in case of G, it is C. So it is showing complementarity to each other. When these two are inserted into this agarobacterium vector or inside the agarobacterium, what happened? This gene 1, gene 2, gene 1 antisense, gene 2 sense, gene 1 producing antisense mRNA, gene 2 produce sense mRNA. Both of are complementarity. So what is complementarity? This showing, in case of A it is U, in case of U it is A, in case of G it is C. So they bind each other. After binding, they forming double-stranded RNA. It is called double-stranded RNA. This double-stranded RNA forming this double-stranded RNA then cut by one of the enzyme known as dicer. One of the enzyme known as dicer. After this enzyme cutting, it is showing a single stranded here. This single stranded RNA, this single stranded RNA or mRNA complex with this mRNA complex with this nematode mRNA complex with nematode mRNA, and this nematode mRNA produced after transcription. After transcription of nematode mRNA, this RNA also present inside the bacteria, inside the bacteria. And when it introduce inside the body of nematode, this gene, this RNA, here is the RNA, this RNA complementarity with nematode mRNA. Due to complementarity, and it shows blockage of this mRNA of nematode. Due to silencing of this nematode mRNA, it not produce protein. Due to no protein, the organism will die. So this is the application of RNAi in RNAi in the